Hello, everyone. I'm your host, Avery Zimmerman, and today's session is the first edition of our Rare Disease Expert Insights webinars, presented by MMS. As an innovative, data-focused CRO, MMS brings insights from our global team of highly respected data and regulatory experts across 4 continents. As a reminder, your microphones will be muted for this session and we'll send out a recording following the webinar. If you have questions, please ask them at any time in the Q&A icon at the top right of your screen. We'll answer them at the conclusion of today's session. With that, I'll turn it over to Dr. Uma Sharma, Chief Scientific Officer.
Thank you, Avery. And thank you all for joining us today for this webinar. When you or your child is sick or have been diagnosed with an illness, the first thing we do is look for therapeutic options, approved medications, side effects, support groups, is the ailment chronic, and many such things. Have you even thought of ailments where you and others may be the sole few contributors to this experience? No approved treatments, some odd stories here and there, and a few anecdotal success treatments along with a whole litany of failures. This is what we're going to talk about today. Rare diseases are something we all have heard about, and few of us know family or patients who suffer from the same. But we rarely put much thought or effort into what goes into developing treatment options for them. Rare diseases are often things you may know about with much research done, like multiple sclerosis, narcolepsy, or cystic fibrosis, based on the incidence in the population, and may also include many N of 3 or 4 in the population at large of genetic etiology. The latter often fall in the category of ultra-rare diseases, and we are very passionate at MMS about our efforts in this area as well. Just last week, we launched our pro bono efforts with regulatory consulting and support for patient advocacy groups and parent groups for ultra-rare diseases. I encourage you to please take a look at that. In this series of webinars that are aimed at understanding rare and ultra-rare diseases, we at MMS make a humble attempt at explaining the regulatory pathways, case studies for various scenarios, specific designations such as ODD, as well as statistical implications of various strategies in the field. It is our hope that as we raise awareness and understanding, the work in the area of rare and ultra-rare diseases will gain more interest and allow for more therapeutic options for these patients. With that, I'd like to introduce a video that talks about a sponsor working very effectively in this space.
Perfect. Well, thank you very much for asking us to be part of the MMS sponsored full webinar on rare disease. It's a real pleasure. My name is Steve Ryder. I'm a clinician by training, have been in the business for over 35 years, held a variety of positions, mostly at Pfizer, Astellas, Alexion, and now great pleasure to be asked by the gentleman on my right, your left, Martin Mackay, to be part of his organization at his new biotech company, Rally Bio. Martin?
Well, thank you, Steve, and what a pleasure to be here with you today. Yes, Martin Mackay, a career in pharmaceutical R&D, most notably at Pfizer, AstraZeneca, and Alexion. And I had the great pleasure of working with Steve at both Pfizer and Alexion. I'm now in this startup, Rally Bio, which we started at the beginning of 2018. And our mission's to transform the lives of patients with severe, rare, and ultra-rare diseases. Pleasure to be here.
So one of the things, Martin, is I was thinking about this for quite a while, just trying to gather my thoughts together. We're in this business some time, and we are really now dedicating ourselves to transformation in rare disease. And really there's 2 reasons that I can think of. The first is that the medical need clearly is still remaining. I mean, I'm not trying to be silly, but of the rare diseases, we can count on a couple of hands those where the transformation has in fact taken place, and we don't have enough hands probably, certainly at Rally or maybe even on this video, for all the rare diseases where the transformation has not yet taken place. So the medical need remains real. And one of the things is that we're going through a period of time when the biology and the clinical path of an increasing number of rare diseases is actually becoming clear enough so that we can really grab it. And a couple of examples are products that we have here at Rallybio.
Yes, indeed. No, I couldn't agree more. Steve, it's a very compelling area to work in. And you and I have worked in big diseases for, most of our career, but 2013, we got together a biotech company who was devoted to this notion of rare disease and transformative therapies. And one of the things that struck me during all that time was the fact that we met patients, we met carers of patients, we met families of patients. And somebody said to me recently, irrespective of the epidemiology of the disease, we gave hope to those folks. And it was such a humbling experience, actually, to work so closely with the families that have those. And so it was a natural progression for us at Rallybio to really continue this fine work and again come up with these transformative therapies. Steve, you could maybe talk about FNAIT. As a great example.
I definitely will. And before that, I'll follow up a little bit on your comment. I mean, one of the things that certainly Martin was my missionary that led to my conversion. And part of the process was exposure to a number of patient testimonials in a bunch of different rare diseases: paroxysmal nocturnal hemoglobinuria, PNH, hypophosphatasia, HPP. There are incredible stories of children whose lives have been transformed by Strensiq. Neuromyelitis optica spectrum disorder, again, young women who no longer have to face this literally a sword of Damocles being held over their head, waiting for a relapse that could potentially leave them blind or paralyzed because of the treatments that we brought forward. Here at Rallybio, we found a couple of diseases where the biology and the clinical paths seem to be coming into alignment. One of them is FNAIT, fetal and neonatal alloimmune thrombocytopenia. This is the platelet version of RHD disease. Most of us are familiar with RHD disease. It's the positive or negative that you're given after you donate blood. I'm B positive, so I'm positive for Rh factor D. And those women who are Rh factor D negative, if they have a child that's RHD positive because of the paternal influence, they get RhoGAM or Winrow during their pregnancy and at the time of birth to prevent what in the past was a true tragedy for neonates. Erythroblastosis fetalis was when it occurred in utero. That same type of disease can occur, but not in erythrocytes, red blood cells, but in platelets. And when it occurs, it can also lead to devastation, just like it did with erythrocytes. We now have the tools to develop for this disorder, essentially the RhoGAM or the Winrow, to hopefully prevent it. It's a disease that we have the biology, we have the technology. Monoclonals are part of what we do. We know the antigen, we know the structure of the antigen, we know that it's a single polymorphism in position 33 of the beta-3 integrin amino acid change. Leucine is A, proline is B. So all of this is worked out, and we're well on the way to establishing proof of concept. Recent announcement this month, July, where we believe we have seen proof of concept in HP1 BB males, receiving HPA1A platelets with a dramatic acceleration of their clearance when you administer the antibody. So this is just one example of what we're doing here at Rallybio. We're certainly engaged. I think we have the tools, technology to meet some of these challenges, and that's what we want to do with basically the rest of our career, I think.
Yes, indeed. And I think it's critical that it's such a good example, Steve. We know the biology. We know that babies are born with this terrible condition, this thrombocytopenia caused by the mismatch in the platelet antigens. And quite common, I've found, in rare diseases, you cannot do this alone. We have to work with exquisite partnerships be able to take these programs forward. And whether it's experts in the field of which we have developed relationships, or whether it's with people that work with us to make sure that we do the best clinical programs possible, is just absolutely key to forwarding these programs. That's why we're so keen to be with you today and be able to say that.
Yes, so that's pretty much what we wanted to say. I think that this is an area that I would invite all of the listeners of this broadcast to please think about, and I hope that we've convinced you that this is well worth your time and the investment of your time, which is the most precious commodity that you have, to try to really make a life-saving, life-transforming difference in so many patients yet to be successfully treated. Thank you.
I wish to thank MMS's founder, Dr. Sharma, and our friends at Rally Bio for helping us kick off this important rare disease webinar series. Today's webinar session will focus on rare disease research in the United States, understanding FDA guidance and flexibility in the application of regulatory standards. Our presenters prepared so much great information on today's topic that we have split the original focus of today into 2 installments. Today we will present part 1 and we'll present part 2 2 weeks from today on Tuesday, August 31st at 2 p.m. A link to the part 2 recording will be provided following the next installment for anyone who attends today's session but is not able to attend on August 31st. Here to present today is Dr. Amanda Biester, Senior Manager of Global Regulatory Affairs, and Ben Casper, Senior Manager of Regulatory Strategy. The topics for this 2-part webinar include a summary of challenges faced in rare disease research, the regulatory framework and built-in flexibility for drugs intended to treat rare diseases, and the emergence of a pathway of treatment of ultra-rare indications. In part 1, we'll cover the challenges and legal framework as well as areas of flexibility in CMC and non-clinical studies. In the second installment, taking place on August 31st, we'll cover flexibility as it applies to clinical development and discuss the path forward for ultra-rare disease research. With that, I'll hand it off to Amanda and Ben.
Thanks, Avery. So first, we wanted to provide a baseline on what it is we are talking about today, rare diseases and some of the associated challenges to research in this context. I won't go into too much detail on this topic as we have the subsequent session to cover orphan drug designations, but the Orphan Drug Act defines a rare disease or condition as a malady that affects fewer than 200,000 people in the United States, or another indication that's unlikely to be commercially attractive to drug manufacturers. One thing to point out here is that many, in fact, most of the approximately 7,000 known rare diseases are much less prevalent than the cutoff used by the FDA, and terminologies such as ultra and uber rare are not standardized by the FDA, nor are they separately incentivized or regulated. On the next couple of slides, I am going to be pointing out some of the challenges of rare disease programs. Keep in mind that for the most part, these challenges pose an even greater degree of difficulty the fewer patients a disease affects. We will also go into some case studies later on, but typically the smaller a disease population, the more regulatory flexibility is applied. So what are some of these challenges to drug development that are frequently more difficult to address in the context of a rare disease? We've outlined them here on the left side of the slide, and I'll go through these one by one. So this first one is centered on the knowledge of the disease itself, which is frequently very limited, both from a scientific standpoint as well as from a regulatory perspective. First, from a scientific standpoint, there is often a lack of understanding around the pathogenesis and pathophysiology, or the mechanistic basis for the disease. This can impede upon the ability to even reach a diagnosis. But there's also often a lack of natural history data around the clinical presentation, the progression, and the resolution of the disease, and even less data on the genotypic and phenotypic variations within the disease itself. In addition, while an animal model may be of particular value, especially for these pediatric diseases, they may not exist, and they can be enormously challenging to develop given the complexity of the disease and the relatively limited understanding of these diseases. And even if they are created, they may not exhibit some of the clinically important manifestations of the disease. There's also frequently a lack of an established regulatory standard. Unlike the more well-studied indications, there is typically a lack of precedence, and there's also no established method for evaluation of a treatment effect. Things like choice of endpoint, choice of assessments, biomarkers, as well as just standard critical elements of a study, study population, duration, and other critical elements are not frequently readily available or predefined. Patients may have a highly diverse clinical manifestations or rate of disease progression that are difficult to predict. And finally, another issue is that the FDA may be changing how they classify the disease, so it can be difficult to keep up with changes in the regulatory landscape as new information about a rare disease or other related diseases or indications comes to light. The second challenge may seem more obvious on its face because it is how we define these diseases, and that is that a small patient population means relatively few patients are available to enroll into the clinical trials. But what might not be as clear is that often these rare disease trials are in very sick, very young populations who have no alternative therapies. As a result, there is a very real and understandable difficulty with early attrition, especially for those not receiving a clinically relevant benefit on this study. And there's this overall hesitance to enroll in any program that has a placebo arm, even if it's a crossover or run-in design, because of the significance of these disease-related events and the potential for long-term morbidity or even early mortality. Next slide. While the Orphan Drug Act has dramatically improved the state of rare disease research in the US, there is still a focus on diseases at the higher end of the disease prevalence spectrum. Diseases such as sickle cell disease or cystic fibrosis still present an attractive profit margin, especially given the ODA framework. Even still, it is not uncommon for even these such programs to struggle initially due to limited funding or resources. As these initial programs may be driven by a scientist physician who has noticed something maybe in the lab or in the clinic, or who has an idea based on the pathogenesis of a specific disease, there may be a limited understanding of the regulatory pathway or the documentation needed at each step of the development process. As FDA incentives apply across the entire breadth of the rare disease spectrum, it seems that the FDA has not found a commercial incentive that consistently compels the pharmaceutical industry to take on the difficult challenges in the setting of a disease that impacts very few subjects. In ultra-rare diseases, the initial drive to find a treatment is frequently entirely owned by a parent or patient advocacy group, often started by a parent. Eventually, the disease or the patients may draw the attention of a physician or scientist. But even with all of the incentives, the initial research on ultra-rare diseases is frequently undertaken by independent investigators or physicians through one of the FDA's programs, such as an emergency IND or expanded access or compassionate use protocol. Due to the lack of experience of some of these individuals, initial studies and the documentation and manufacturing that is typically prepared at the earliest part of the development may not meet the regulatory expectation, which can slow or alter the typical path toward development of an experimental treatment. Note that in these instances, much if not all of the patient population may receive the therapy in the research setting anyways, and that licensing actually becomes extremely rare. Next slide, please. The last challenge I wanted to talk about today is one of the same issues that has historically impacted standard drug development as well. In this case, I'm specifically talking about a lack of harmonization. There's no universal definition of a rare, ultra-rare, or neglected disease, or of the treatment options in development for such diseases. They're differently incentivized, differently considered within and across different sectors, such as health authorities, insurance providers, regulators, researchers, and patient organizations. With that, I'll turn over to Ben.
Hi everyone, Ben here. Really excited to be here to talk to everyone. What I'm going to do is just go over some of the basics in terms of the legal framework for rare disease research. So we're going to start with a quick overview in the next few slides. So to really understand the history of the orphan development program in the US, it is extremely helpful to actually go all the way back to 1963 and the Kefauver-Harris amendments. These really ushered in the modern era of the FDA through the requirement for demonstration of clinical efficacy. So what we think of as the modern phase 3 trial in the standards surrounding their design, execution, and analysis really grew out of these amendments. So while Kefauver-Harris revolutionized the regulation of drugs, in some ways, it also created barriers for the development of drugs for rare disease, namely around the difficulty and the expense of demonstrating efficacy in small patient populations. By the 1980s, it was pretty clear to Congress and regulators that it was in the public interest to reduce the cost of developing such drugs and to provide other incentives that would allow the development of drugs for rare disease to proceed while critically retaining the existing statutory standards for safety and efficacy. So to achieve this objective, the Orphan Drug Act, or ODA, was initially passed by Congress and signed into law by President Ronald Reagan in 1983. So this law created both a legal definition for rare disease and financial incentives for drug and biologics manufacturers to develop treatments in this space. The definition and benefits will be covered in detail in a subsequent section. In addition, the Office of Orphan Product Development, or the OOPD, was also formed in 1983 and was tasked with administering the provisions of the ODA. The OOPD has 6 core programs, 3 designation programs, and 3 grant programs, which again, my colleagues will outline in more detail in the next session. Similar rules governing medical devices were passed in 1990. In 1991, the regulation was proposed to codify existing administrative practices that had implemented the ODA and its subsequent amendments. The result of that was the Orphan Drug Final Rule, which was codified in 1992. This rule was actually again revised in 2011, and the final rule incorporating those revisions has been in effect since 2013. Essentially, those amendments were intended to clarify, streamline, and generally improve the designation process. Of particular note, in 2012, a designation was made available for rare pediatric diseases, which was in line with other initiatives put in place to encourage the development of pediatric therapies. More recent developments include the Rare Disease Cures Accelerator, which was begun by CEDAR in 2019. The accelerator is currently being developed as a data analytics platform, standard core set of clinical outcome assessments and endpoints, and a clinical trials network for rare diseases. Lastly, as of last year, the FDA announced their orphan drug technology modernization effort and opened an online portal for the submission of new orphan drug designation requests, further easing the burden on potential drug developers for rare diseases. And while today's talk is focused on US regulatory, in the FDA in particular, the ODA inspired implementation of orphan legislation worldwide, with regulators in Japan, Australia, and the EU following suit throughout the 90s. One take home from the history of rare disease regulation is that the Orphan Drug Act represents a means of expanding the practical range of Kefauver-Harris. The incentives do not change the statutory standards for drug approval. Rather, they create a structure wherein those requirements might be met in a manner that makes financial sense for drug developers considering enrollment of a rare patient population in clinical trials. What that means is that sponsors must still provide evidence of the drug's effectiveness for its intended use from adequate and well-controlled studies, and they must provide sufficient information for the FDA to conclude that the drug is safe for use under the conditions prescribed, recommended, or suggested in the approved labeling. So while the ODA represents one expansion of the practical application of Kefauver-Harris, the other major related expansion is the FDA's expedited programs for serious conditions, which create incentives for the approval of treatments for indications associated with morbidity that has substantial impact on day-to-day functioning. So while the seriousness of a disease or condition does not have to be demonstrated to obtain orphan drug designation, most rare diseases are serious. Thus, the FDA's expedited programs for serious conditions typically inform the regulatory perspective on orphan drugs. And I'll illustrate this a bit in the next slide. So in 2020, 31 of 53 novel drugs approved by CDER had an orphan designation. These 31 approvals are listed here. Orphan drugs that also utilized one of the FDA's expedited programs for serious conditions are shaded in gray. So what you can see here, and this is pretty striking, is that out of 31 novel orphan drug approvals, all but 2 utilized at least one of these programs. And the 2 that did not have a designation and use the standard review pathway were actually also for serious conditions. So those include Listerisa for Cushing's disease and Sarclisa for multiple myeloma. So if the legal framework defined by the Kefauver-Harris amendments does not change, what does change when we talk about strategies for the approval of orphan drugs? In short, it's the therapeutic context that changes. The therapeutic context really impacts the assessment of risk-benefit that the modern FDA uses as a structural framework for approval decisions. In that regard, both the severity of the disease and expected benefit of the therapy, or how it addresses a medical need, directly influence the acceptability of the risks of a therapy. And when it comes to serious diseases or conditions in general, the FDA broadly interprets the statutory requirements to the extent possible whenever data on a particular drug are convincing. Thus, for example, the FDA has historically allowed for reliance on only a single adequate and well-controlled efficacy study to support approval in instances where the sponsors are able to provide strong evidence of an important clinical benefit, and when a confirmatory study would be impractical and/or unethical. So what does this really mean for rare disease research? The statute generally allows for flexibility in applying regulatory standards and specifically states that it allows for the exercise of scientific judgment in the kinds and quantity of data required for a particular drug for an indication. For rare diseases, this will be influenced by the size of the disease population and the seriousness of the indication, as well as the presence of other extenuating circumstances or complicating factors pertaining to the population or the disease or condition. In addition, the OOPD encourages innovation when it comes to study design in an attempt to overcome or work with the challenging circumstances that rare diseases can present. For its clinical study grants, the FDA is encouraging innovative methods such as adaptive trials, which allow for planned modifications to one or more aspects of the design based on data collected from the study subjects while the trial is ongoing, seamless trial designs, which compress the phases of a trial into one continuous trial, and basket, umbrella, and platform trials, which allow for testing of multiple drugs and/or multiple diseases using a common infrastructure. These types of studies are given priority during the review process for grant applications. And finally, while there may be some hesitance on the part of regulators to move away from the gold standard of randomized controlled trials, OOPD is also actively encouraging the use of modeling, simulation, and real-world evidence. So modeling and simulation are critical tools for evaluation of new developments in small populations. Because of this, the OOPD also gives priority to natural history studies if they have potential to exert a broad impact across multiple rare diseases and if they plan to use simulations and modeling in conjunction with the data from the natural history study. The OOPD has also forged ahead with the use of real-world data to better understand patient populations and their needs and to improve outcomes by allowing for more efficient design and conduct of clinical trials in the healthcare setting to answer questions previously thought infeasible in traditional clinical trial designs. So we're going to present a number of case studies today, and they're going to follow a similar format where we'll introduce you to the key concept, summarize the therapeutic context and physiology, and then provide a high-level overview of the regulatory setting at the time of the FDA's review. Finally, we will discuss the outcomes of the FDA review of a particular application and how it might be tied to the rarity of the disease. So we'll talk about therapeutic context in this first case study. To illustrate the critical importance of therapeutic context, we're going to look at this recent approval where the FDA allowed for reliance on a single, adequate, and well-controlled efficacy study in order to identify some of the factors that may have contributed to this decision. This is a case study for Givlaari, which received approval in 2019 based on evidence from a single trial. As we are going through the case study, note the therapeutic context in combination with the strength of evidence of a treatment effect. So first off, let's get into the therapeutic context. The rare disease that was pursued here is acute hepatic porphyria, or AHP. It's actually a group of 4 genetic diseases caused by increased aminolevulinic acid synthase, or ALAS1 activity, which results in increased levels of neurotoxic heme intermediates. The symptoms of AHP often resemble those of other gastrointestinal, gynecological, and neurological or neuropsychiatric diseases, and vary widely across the patient population. These symptoms typically include both chronic symptoms and acute attacks, which are frequently characterized by severe abdominal pain and other symptoms. These severe symptoms can lead to hospitalization, irreversible neurologic damage, and increased risk of hepatic malignancy. So right off the bat, we see this as a serious progressive disease with significant and sometimes irreversible morbidities. The second consideration for therapeutic context is the availability of current therapies. Prior to the approval of Givlaari, management of these attacks often required hospitalization and supportive care. There was one FDA-approved medication, Heman, but this could only be used in a subgroup of patients with one of the 4 genetic diseases. If the attack was related to the menstrual cycle, a Heman infusion could be administered if available, but this was not a commonly stocked drug, and access was frequently delayed. Givlaari is a small interfering RNA or siRNA that inhabits aminolevulinic acid synthetase 1. At this time, this application got a lot of attention because it was the world's first approved RNA interference therapeutic. So we can see from the previous slide that Givlaari, as a candidate, is very much in line with the 31 FDA-approved orphan drugs we reviewed previously. That is to say, it is a treatment for a rare disease that is also a serious condition for which satisfactory therapies do not exist under the current standard of care. Now that we understand the context the FDA had for weighing the benefits and risks of Givlaari, let's review the safety and efficacy evidence submitted for approval. First, this was a single study. It was an adequate and well-controlled multi-center trial that was able to provide strong evidence of an important clinical benefit. In this case, the data speak for themselves. Efficacy was measured by the rate of porphyria attacks that required either hospitalization, an urgent healthcare visit, or an intravenous Heman administration at home. The sponsor reported an estimated attack rate over the 6-month double-blind treatment period of 2 attacks per patient in the Givlaari arm, compared to 14 attacks per patient in the placebo arm. This is 70% fewer porphyria attacks with treatment, or approximately 10.4 fewer attacks per year. In addition, Givlaari is administered subcutaneously, offering adult patients with AHP a prophylactic therapeutic option which potentially may be more conveniently administered compared to heman. So, given this information, what was the likely outcome and how is it tied to the rarity of the disease? So just to recap, this is a treatment for a rare disease that is also a serious condition for which satisfactory therapies do not exist under the current standard of care. And as we have seen, a strong treatment effect demonstrated in an adequate and well-controlled multi-center trial. So, combined with a strong safety profile, the FDA determined that the benefit-risk analysis favored the approval of Givlaari for the treatment of adult patients with AHP. No advisory committee meeting was held, but the FDA did hold the sponsor to one post-marketing commitment, a controlled trial in adolescent patients with AHP. So, in terms of key takeaways, one key point: sponsors need to understand how precedent, regulations, therapeutic context, and overall risk-benefit all factor into the approvability of orphan drugs. Many sponsors may assume NDAs for orphan drugs only require one study. While this is often true, it's not always the case, and what we want to clarify is that it's really the combination of the seriousness of the disease, the lack of availability of adequate therapeutic options, and the presence of a dramatic treatment effect demonstrated using a clinically relevant endpoint that typically allows the FDA to exercise their scientific judgment. In this case, it's not simply the fact that AHP is a rare disease, it's all of those factors combined. Leeway on the part of the FDA is considered on a case-by-case basis and should not be considered a standard assumption. Next, we'll go into some more detail on how flexible regulatory standards apply to the different disciplines of drug development. For this section of the presentation, we will review some of the guidance the FDA has produced for the industry in different disciplines. The list of applicable FDA guidances goes well beyond the scope of this webinar. However, there are many more guidelines and guidances that may be particularly useful and should be referenced when communicating with the agency on these topics. Also, note that we have not included any guidance on the designation or grant programs offered by the OOPD, meetings with the OOPD, or on expedited programs for serious conditions, as these will all be the focus of the second webinar in this series. So the FDA explains their thinking on common issues in drug development that are frequently more difficult to address in the context of rare disease in a draft guidance for industry that was most recently revised in January of 2019. This guidance states, and I quote, "The flexibility extends from the early stages of development to the design of adequate and well-controlled studies required to demonstrate effectiveness to support marketing approval and to establish safety data needed for the intended use." So today we're going to really look at some of the key points and areas of flexibility. Specifically, first I'll discuss areas where the FDA may be flexible on the type and extent of required manufacturing information. Factors that the FDA evaluates when determining areas of non-clinical flexibility, and factors that the sponsor should consider when determining the relevance of existing data. After this, Amanda will discuss the FDA's recommendations for generation of clinical data in rare populations, including their thoughts on trial design, standardization of data collection and handling, adequate effectiveness and safety data, use of historical controls, and early randomization of natural history data. Okay, so the common issues in rare disease guidance addresses the importance of drug manufacturing considerations in drug development programs for rare diseases. In a typical drug development program, drug manufacturing will undergo development concurrently with clinical development. With each stage of development, agreement is reached on the requirements for the next stage of development, whether that be preparing for the first-in-human testing, preparation for phase 3 clinical trials, or scaling to the commercial formulation. This particular guidance focuses on 3 main topics: changes to the manufacturing process and or formulation, type and extent of manufacturing information, and late-stage modifications to manufacturing procedures and purification methods. The first is the importance of careful assessment of planned formulation or manufacturing changes at any phase of development to determine if the changes could directly affect the safety or efficacy of the product. The second is the type and extent of manufacturing information that is expected at the time of submission and approval for certain components. Some examples of this type of information could include stability updates, validation strategies, inspection planning, or manufacturing scale-up. The FDA has stated that they will explore the level of flexibility on a case-by-case basis. The 5 factors they will consider are listed here, and those include product characteristics, seriousness of the condition and medical need, manufacturing process, robustness of the sponsor's quality system, and the strength of the sponsor's risk-based quality assessment. Finally, although not directly stated in the guidance, the size of the population to be treated, both in clinical phases and during commercial manufacturing, will be taken into account. The third topic is frequently of particular relevance to rare disease programs, and that is scale-up, which can result in late-stage modifications to manufacturing procedures and purification methods. A word of caution here, even minor changes to manufacturing or purification processes can result in unexpected changes to drug characteristics. It is not, for example, uncommon for the responsibility of manufacturing to change hands during a drug development process, but this is an especially common situation when it comes to orphan drug products. For example, products might change from a single investigator or patient advocacy group to a company, or from a small company to a large company. If initial studies are not being done with a high enough quality product, all of the initial work can be negated by these unexpected changes, which then results in the need for additional non-clinical testing, and in some cases, even additional clinical trials. For this reason, inexperienced researchers should consult with the agency guidance and with the FDA themselves to understand whether or not their drug product characteristics are adequate for commercial scale-up, and if not, to ensure that the drug being used is appropriate to the level of development and is not likely to cause further issues down the road. A second guidance I wanted to review in the context of quality is the guidance on pharmaceutical development, which describes the principle of quality by design. So quality by design principles focus on building quality into the product and take a systematic approach to development, beginning with predefined objectives. Because quality by design can lead to a higher level of product and process understanding, it contributes to a higher likelihood of success in the overall development program. This increased process robustness can contribute to a greater potential for regulatory flexibility, easing any need for process changes and possibly allowing for flexibility for real-time release testing, continuous manufacturing, and other more complex approaches to quality. It may be a stretch for companies to invest the considerable upfront time and money required for quality by design-based manufacturing processes. However, even incorporating some of the elements of quality by design can strengthen the overall development strategy, and thus these principles are worth consideration. So with that, we'll move into non-clinical studies. So shifting focus to non-clinical development. Non-clinical studies are a mandated part of drug development per statutory requirements. The purpose of these investigations typically falls into one or both of 2 categories. That is, either to provide evidence that a drug is reasonably safe under the conditions proposed for clinical investigations, or to further develop understanding of the drug itself, its metabolism, pharmacokinetics, mechanism of action, and potential for efficacy. So as we saw with CMC, there is some flexibility with non-clinical programs as well. Regulations mandate that the FDA exercise the broadest flexibility in applying the statutory standards while preserving appropriate guarantees for safety and effectiveness for drug development in serious and life-threatening diseases. Modifications are even more likely to be allowed for serious or life-threatening diseases where there are no current treatments or where they are inadequate, the FDA has stated that this flexibility applies to the non-clinical data needed to support a clinical development program. So we'll look at some additional factors on the next slide. So the factors you see here will be evaluated by the FDA in order to determine how flexible they can be on a particular program. They include the pharmacological and chemical characteristics of the drug, the design and objectives of the proposed clinical investigation, anticipated risk to humans, and the existing accumulated non-clinical toxicology and human data. Thus, the characteristics and prior knowledge of the drug, the clinical study design being supported, and the anticipated risk to study subjects will all be weighed in the agency's decision-making process. The FDA may allow other strategies for ongoing non-clinical development. For instance, they may allow for concurrent development alongside the clinical trials or in a staggered fashion, such that the resulting data from non-clinical studies are submitted before dosing of any patient in an ongoing clinical trial that exceeds the duration of the available non-clinical data. Also, sometimes they'll allow for a delay in the submissions of specific non-clinical trials from the IND to the marketing application, or even allow for a deferral until post-marketing. The FDA has stated that the factors reviewed here should be evaluated when determining the relevance of existing data to support clinical development. The FDA may require additional non-clinical studies if the drug product, formulation, route of administration, or dosage is different, especially when there is any increase in anticipated risk as a result. For example, if dosage is increased above the no observed adverse effect level tested in an earlier program. If there is sufficient justification and when no specific safety concern is present, clinical trials are likely to be allowed based on modified or minimal non-clinical development programs. So having reviewed these areas of flexibility, we'll go into another case study. So for this case study, I'm going to review how label extension can work in the framework of rare diseases and how relying on existing safety data, one of the things we talked about in the review of clinical flexibility, that is existing safety data from other indications can serve as supportive evidence of safety. So here we'll be talking about Kineret, which is an interleukin-1 or IL-1 receptor antagonist, which was initially approved for rheumatoid arthritis in 2001. And that approval was based on a conventional large clinical development program encompassing 3 randomized efficacy studies enrolling a total of 1,790 patients. This initial application demonstrated that Kineret was generally well tolerated and post-marketing safety demonstrated an acceptable safety profile. Since that time, Kineret has been studied in a number of indications. The approved uses vary globally, but we want to focus on here is those approved indications that were given orphan designation in the US during the development program. The first of these is neonatal onset multi-symptom inflammatory disease, or NOMID. NOMID is a lifelong and severely debilitating disease associated with an overproduction of IL-1. And prior to this particular approval, the Kineret approval, no adequate therapy existed for these patients. Untreated patients developed progressive hearing and vision loss, variable degrees of cognitive impairment, and joint contractures. And again, as with the other cases we've reviewed, this is a rare and serious disease. So it's really the therapeutic context that changes the calculus for evaluation of risk-benefit. The second orphan indication is deficiency of IL-1 receptor antagonist, or DIRA. DIRA is an ultra-rare autoinflammatory disease that can occur in the first weeks of life and may be fatal. DIRA is caused by a genetic mutation in the gene encoding the IL-1 receptor antagonist protein. In patients with DIRA, the deficiency of the antagonist protein leads to unopposed action of IL-1 signaling, resulting in painful and life-threatening systemic inflammation with skin and bone involvement and stunted growth. Again, there was no adequate therapy for these patients at the time of the evaluation of the Kineret label expansion. As in the previous case studies, These indications qualify for breakthrough designation, and in this case, priority review. The first application for label expansion was based on an open label study, and in that one, there were 43 NOMID patients, which demonstrating a mean decrease in disease-specific diary symptom sum scores. For the 11 patients who went through a withdrawal phase, disease symptoms and serum markers of inflammation worsened after withdrawal and promptly responded to reinstitution of Kineret therapy. Treatment of NOMID patients for 5 years with Kineret demonstrated that in addition to controlling the daily symptoms of the disease, such as fever, rash, headache, and joint pain, important CNS functions such as hearing and vision remained stable and did not progress on treatment. The second application was based on data from only 9 patients with DIRA treated for up to 10 years in a long-term natural history study at the NIH, in which all 9 patients achieved inflammatory remission well on Kineret. So, given all this information, what was the outcome and how was it tied to the rarity of the disease? Keeping in mind the differences in the number of patients enrolled in these two different populations. So as in the first case study we saw, both indications were approved with no advisory committee meeting required. In the instance of these label expansions, there were also no additional post-marketing requirements imposed. And just to reinforce my earliest point, the orphan indications are the only approvals allowing for use of Kineret in pediatric patients. This is likely due, at least in part, to the therapeutic context presented in these very serious orphan diseases compared to juvenile rheumatoid arthritis. So these approvals highlight the importance of leveraging existing data to support label expansion. In this case, the second label population was based on data from only 9 patients, but relied on a comparable safety profile observed in the 42 NOMID patients evaluated in support of the first label expansion. Thus, the key takeaway here is to really understand the relevant precedent for what existing data can support in the context of additional related rare disease populations. And with that, I'll end the talk, and thanks everybody for your time today.
Thank you, Amanda and Ben, for your insights. Now we'll be moving forward with the Q&A portion of the webinar. I'll hand it back to our presenters.
Thanks, Avery. So we do have a couple of questions. I'll go ahead and read off this first one. In 2020, I tried to submit an ODD to the electronic FDA portal via FDA Form 4035 without luck. I was unable to successfully attach figures and tables to the Word file. Has anyone had success with an electronic ODD submission via Form 4035? If so, are there any tips for success with this form? Ben, do you want to take this one?
Yeah, so I have to be honest with you guys. I haven't used the portal yet. As long as the FDA has that little yellow box on the website indicating that we can use email throughout the COVID, I've just been sending everything to OOPD via email. And part of the reason for that is because as a CRO, the portal is a little bit challenging in the sense that you have to open a sponsor-specific entry. So as you can imagine, there's a lot of logistical challenges there that I think we're still kind of working out, but should have worked out pretty soon. A few things to suggest. It's possible that there were some issues just with the format there. Maybe taking the 4035 and just attaching the figures and tables as a PDF to that might be helpful. Another thing you might want to check is your version of Adobe. I know sometimes with the older versions that form won't work for all of the fields, so could be a software challenge there. It could be just a matter of trying to use Word files in general, and those portals can sometimes be a challenge. But I would say if you do have any issues there, just immediately contact OOPD. They're usually very, very responsive about things like that. And I know that they're actively trying to get sponsors to use that portal. And I think it is going to be a good thing just to have all your sort of non-IND submissions tracked in one place. Ultimately, it's going to be really nice. But I don't have a specific answer there, unfortunately, but I would just say if with those kinds of things, just contact OOPD right away.
Okay, and the second question, do you have any more information on FDA acceptance of real-world data in an ODD application? My understanding of the FDA's ODD webinar is that when establishing prevalence, literature must be submitted with claims database data. My understanding is that FDA will not accept claims database data alone for population estimates. I think that it's not necessarily that they won't accept claims database data alone. It's that you have to show that you've made a good faith effort to find prevalence data, and that claims databases have to be validated. The data has to be validated. You have to also show that it's generalizable to the US population. You have to show the FDA that you recognize its limitations. And also you have to be very careful that you're not submitting data for a subset when you're going for, orphan designation for the whole indication. So I think there's a lot of caveats around when you can use that type of data or how you should use it. And it's true that you can't always rely on that data, especially not just that data. But we do have another presentation coming up on validation, and they will be talking about real world data. So that will be another installment. Okay, so that looks like we've answered the questions. We'll turn it back over to Avery.
Actually, I had another one I just wanted to go through really quick, which is, under what circumstances should a sponsor request a meeting with the OOPD versus their specific review division? And that's a great question. And what I would suggest is that the answer to that question is actually inherent in the discussion from today. That is to say, if the statutory requirements don't change with the orphan application, then the things that you would specifically discuss with your review division aren't changing either. So what I mean by that is, if you have questions around study design, even though those questions might be specifically related to a study that's issues that are unique to an orphan application, they're still study design questions. So you'd still ask them to your review division. You can request OOPD representation on that meeting, but it should be a meeting with that team. That is to say, the team within your division. So another way to think about that is, only questions that are specific to the orphan application itself should go to the OOPD. So if you have a question of prevalence, subset, things of that nature, those should go to the OOPD. In addition, any questions about the benefits of the designation should also go to the OOPD. But outside of that, everything else you'll want to contact your review division, set up your type A, type B, type C, just as you normally would, but request additional input from the OOPD as necessary.
Okay, and one last one came up. You mentioned challenges with early attrition and placebo arms. Does FDA make any recommendations for addressing this when they do require a placebo arm? So I would start out by saying that our experience with the FDA and orphan drug designations has led us to believe that the FDA is very motivated to help, especially when it comes to designing the type of trials that will result in data that they can accept. They do, in their guidance document for rare diseases, they do talk about different ways that they feel that sponsors can help address these. Those include directly engaging with the patient and caregiver groups, like the patient advocacy organizations, to make sure that before you even start a trial, that you are on the same footing and that you might be able to avoid undermining trial recruitment that way. Modified trial designs are a big way. I know I mentioned that sometimes even those don't work, but that's another big way that FDA pushes sponsors to deal with some of these, like I already mentioned, crossover, I think, and run-ins, but there's also dose response, delayed start, randomized withdrawal. There's a lot of possibilities to explore there. And again, this is a great opportunity to engage with the FDA and make sure that you are designing a trial that has its best foot forward as far as recruitment and retention goes. Okay, so with that, I will turn it back over to Avery.
Thank you. If you have additional questions on what you heard today, please follow up with your point of contact directly or email media@mmsholdings.com and we'll direct you to a presenter. Make sure to turn in on Tuesday, August 31st for the second installment of this topic. Following part 2 of today's presentation, the next edition of our rare disease series will take place on October 6th and will focus on orphan drug, rare pediatric disease, and expedited program designations, FDA incentives to promote rare disease drug development. To register for the next rare disease webinar and for additional free webinars, visit mmsholdings.com/webinars or follow us on LinkedIn for updates. Thank you very much for joining us today.